All right, you all, thank you all for coming today. We are continuing our colloquium today with Alex Klotz from the Department of Physics and Astronomy. Thank you for making the drive all the way out here from where you're from. So without any sort of further delay, Alex, go right ahead.
Can I send you my mileage receipts or?
I don't think the department can afford it.
All right, thank you. So the backstory is that a few weeks ago, I gave a talk at a different math seminar. Because I went to the effort of putting together a math talk and practicing it and everything, I thought you guys want to give me advice. So that's if there are any vacancies in the schedule. I'm happy to be here all the way across the quad. So I'm going to talk about, let's see, mathematical investigations of kinetoplast DNA. Assume most people don't know what that is, so I will explain it. Slide, please. All right, so just a brief personal background. I was, okay. Well, so I primarily like in experimental physics. I did my training in a field that uses DNA to study polymer physics. And as a postdoc, I started doing experiments with knotted DNA molecules. So we can see like a knotted molecule jiggling around and untying in that animation. And that's what got me thinking about these more like topology related questions. And then when I started my faculty position in 2019, my plan was to base my lab around kinetoplast DNA. And that was in 2019. So in 2020, you all know what happened. Because I couldn't go to the lab and like my students had limited time in the lab, I started thinking more about computational and mathematical stuff. So the types of things I'm interested in are questions related to topology and biopolymers that kind of relate to the experiments that I do. So interested in knots in open curves, which apply to either DNA or protein. And then what's called the rope length problem, which is just asking how tight you can make a knot. And also topologically linked networks, which is what I am talking about today. So I still consider myself like an experimentalist. I just now feel like I'm doing experiments with mathematical objects instead of physical molecules. So first I'll explain what kinetoplasts are and briefly about the things that I've done with them. So there's a family of single cell parasites called trypanosomes. They cause a few tropical diseases in humans. So Trypanosoma brucei causes African sleeping sickness and it's transmitted through the tsetse fly. And then Trypanosoma cruzi causes Chagas disease. And then there's various species of leishmania, which cause leishmaniasis through sandfly bites. So what these have in common biologically is like what looks like an eye in these microscope images, except they are single cell organisms. They don't have eyes. So these are actually dense clumps of DNA, and I will explain what those are. So just to briefly explain how biology works. So DNA has a certain genetic sequence. It's a fairly inert molecule, but it gets copied into RNA, which has a pretty similar alphabet and is much more chemically active. The RNA gets fed through a little molecular machine called a ribosome, and every 3 letters of RNA recruit an amino acid to a growing peptide chain, which eventually folds into a protein and goes on to do the chemistry of life. So with trypanosomes, if you look at their mitochondrial DNA, it's circular, just like ours. And in this context, it's called a maxi circle. And due to some ancestral mutation, it's said to be encrypted and that it doesn't code for a properly functioning protein. So instead of just dying, they developed this way to edit their own genes. They have these other loops of DNA called mini circles, which are smaller. They get copied into what's called a guide RNA. And they're able to add and remove uracil from the mitochondrial messenger RNA, like in this artist impression. And that allows like the proteins to be expressed, or the genes to be expressed, the proteins to be transcribed, translated, whatever. And then, the parasite can go on to metabolize. So each parasite has many different kinds of mini circle that edit different genes on the maxi circle. and the cell needs all of them to survive. So when the cell divides in two, it needs to make sure that each copy gets all the mini circles it needs. And instead of relying on Brownian diffusion to do this, it keeps them like topologically linked in like this medieval dungeon keeper keychain structure, and then cuts that into when the cell is ready to divide. So this DNA structure is called a kinetoplast. It's best described as DNA chainmail, kind of keeping with that medieval theme. It's made of about 5,000 of these mini circles and a few dozen of these maxi circles, which contain the mitochondrial genes. And inside the cell, it looks kind of like a hamburger. If you remove it and isolate it and look at it with some kind of high resolution microscopy, you can see like some very intricate structures where the different molecules meet, and you can image it down to the level of the individual topological links. So when we talk about the topology of kinetoplast DNA, that's usually referring to like the network structure of like which mini circles and maxi circles are connected to which other ones. And this is usually represented as a graph. So each mini circle is represented as a node in a graph, and if they share a topological link, they're connected by an edge. So there was an experiment done in the 90s where they used some enzymes to break up kinetoplast networks, and then ran the like chopped up DNA through a gel and looked at how it split into like different bands corresponding to one loop or two loops or three loops. And by looking at like the ratio of double loops to single loops or triples to singles as a function of how much of the DNA they cut, they can compare that to what different like lattices predict, just with some basic like graph theory. And the conclusion was that each mini circle is linked to three others on average. So it's said to have a valence of 3. And the lattice that was most consistent with the data was the honeycomb. And I think the idea of trivalence has stood the test of time, but everybody thinks that the honeycomb picture is too simple, and I will come back to that. My tool as an experimentalist is mainly fluorescence microscopy. So looking at kinetoplasts in a microscope, each one of these little blobs is like a kinetoplast made of 5,000 smaller loops of DNA. Before I started doing these experiments, I was kind of picturing in my head like some kind of like fuzzy, like, I don't know, DNA spaghetti monster. Here's a picture of about 100 of them and a movie of one rotating in 3-dimensional space. You can see they all have fairly common sizes, but very different shapes. Just highlighted like from a different experiment, like a chubby starfish and a backwards 4 and a flying saucer. And most of them are the same size, but if you look at this one, and it's hard to control the mouse on zoom, and that one, those are about twice as big because the cell has basically doubled them before it gets split into. So we can also use confocal microscopy to image them in 3 dimensions, where we see they have this positive Gaussian curvature. I describe them as like wrinkled hemispheres or like 5-micron fluorescent jellyfish. The double-sized ones are a bit flatter, and again, this like positive curvature wasn't really appreciated before we started doing these imaging experiments, and I'll talk more later about where that might come from. I also have a student over in the physics building doing some very nice atomic force microscopy images, where basically you drag a very like sharp tip across a surface and look at the interactions between the tip and what's on the surface. And we can see sort of the individual links between molecules, and just make some very nice pictures. And although we have all these nice pictures, we don't have much in the way of analysis yet. I'll talk about some other AFM analysis later, but for now, just look at these nice images. All right, so most people, including biologists, haven't really heard of these. So when they hear about them, they get some fairly I get questions, most commonly like various forms of what the hell. So this isn't really a biology crowd. So I will just leave this up here while I talk and now move on to the next slide, but I'm happy to answer whatever questions I have about the biology. All right, so like the main reason to study these, I guess, on a societal level is to like prevent and diagnose and treat parasite infections. So these are an appealing target for drugs because humans do not have kinetoplasts. So if you have a drug that disrupts this mechanism in some way, then you could give it to somebody and kill off the parasites within them without harming the human. So there's a drug called Baranil, which does this. I think it's mostly given to horses, but actually don't quote me on that. But it's known that if you look at the kinetoplasts of Trypanosoma cruzi, which causes Chagas disease, it's been treated with this. It changes the topology of the kinetoplast network. So if basically you get these like fibers of excess DNA linkage forming within the kinetoplast, that kind of increases as you give them more and more of this drug. So this could kind of suggest a future connection between this kinetoplast biophysics that small number of us are doing and the broader treatment of parasitic diseases, but we are not there yet. My focus was more on material science. So I started like doing research in a field that uses DNA molecules to study polymers because you can see things in a single DNA molecule with a microscope that you can't see with like synthetic polymers, and you can learn about like dynamics on a molecular level. So in the past couple of, let's say, decades, there have been kind of two emerging classes of material. There's been two-dimensional materials like graphene, which are mostly developed for their potential electronic properties. And then various forms of like topologically complex molecular architectures. That means like polycatenane, rotaxanes, molecular knots and things like that, which are like candidates for like, chemically powered nanomachines and other materials with exotic properties. And then there's the occasional material that people develop that combines both. So there was like an article in Science a couple of weeks ago where they made these two-dimensional, like topologically interlocked networks. So just like it's difficult to study polymer physics on a single molecule level unless you use DNA, it's hard to study what's happening at the molecular scale with these like complex molecules, or if you call it graphene cheetah molecule. So we'd like kind of a mesoscopic system where we can see, like for example, how do thermal fluctuations affect a two-dimensional molecule as it fluctuates into the third dimension. So kinetoplasts are also like in the center of this Venn diagram. I guess this is the math department doctor called an Euler diagram. So the idea would be to use these as like a model system to study both two-dimensional and topologically interlocked materials. So kind of my first project on this, which was my last year as a postdoc, was just characterizing the equilibrium properties of kinetoplasts like in solution. What are their, how do they fluctuate, what time scale, how do they deform and restore from deformation. And then when I started my position here, I was interested in various forms of destructive testing. So for example, if you remove 10% of the mini circles, do you change the Young's modulus by 10%? I don't know the answer to that question, but it's kind of going in that direction using either light or enzymes to degrade the DNA. This is actually a paper that Ryan was a co-author on. Where was I? Oh yeah, so the challenge slash opportunity when you start to remove mini circles from these networks, like is happening in this animation, is that eventually you no longer have a network and the whole thing just explodes into like this cloud of formerly linked DNA. So this is related to the concept of percolation. I don't know if everybody knows about percolation or nobody does or somewhere in between. I guess kind of like a percolation problem. So I'll just give a brief overview. So if you have like, say, a network consisting of nodes with a certain amount of connections between the nodes, there's a certain amount of connections you can have where there is like a single component that spans the entire system. So when you hear this word outside of like science or math, it usually refers to coffee, where you have a percolating network of pores between the coffee grounds that allow water to make contact with all the grounds. So in the context of kinetoplasts, basically you can remove mini circles from these networks, and when you sort of the density of links gets below the percolation threshold, you no longer have a network, you just have a bunch of different DNA molecules. So there's different experiments that can be done like above or below the percolation threshold. Above, you can study like, how two-dimensional objects behave in a three-dimensional fluid. Below, we can look at like these more individual topological links and like try to stretch them and measure the like force response of that that interlock bond. And right at the percolation threshold, you get these kind of fractal structures that are that transition between being like this one-dimensional branch polymer and a two-dimensional sheet-like polymer, which is cool, but hard to attain. So that is kind of my introduction of what kinetoplasts are and what I was doing with them. Now I'll talk about two different more mathematical projects related to them. So the first I will call the topology curvature relation in chainmail networks. And I think the images kind of tell you where I'm going with that. So looking at these things, a kind of question that comes up is why are they curved? There's no like obvious biological reason, and you can make various like plausible sounding physics-based arguments for why they should have positive Gaussian curvature. I think the actual answer comes from this like kind of this region of extra linkages around the edge of the networks, which you can think of like the elastic band in a shower cap, which keeps the whole thing under tension, which is resolved by curving the network. There was actually like a preprint that came out yesterday also talking about this, which I have not yet incorporated, but doesn't really matter because I'm not talking about that anyway. So going back to 2020, when our lab was shut down, I had some students who needed to continue their research, and one of them, Edgar Garcia, just wasn't able to return to the lab. So in collaboration with James Polson and Prince Edward Island, we undertook the first simulation of kinetoplast DNA, at least on a network scale. So we simulated what's called, well, I now know it's called Japanese chainmail, which has like a lattice of planar circles, each connected to their neighbors by perpendicular circles. And we use this Monte Carlo to just randomly translate and rotate the individual rings. So each of these mini circles is represented as a rigid circle, which is like a rough analogy. And we just had a criterion that any move that preserved the topology was accepted and any move that changed the topology was rejected. So what we were looking for was like, how do the eigenvalues of the gyration tensor depend on molecular weight, which is like a thing polymer physicists care about, but we found something more interesting than that. We did get good eigenvalue results though. What we noticed was that the networks, when they reached equilibrium, had the same positive Gaussian curvature that we see in biological kinetoplasts. And this was something that we saw with a few different like types of lattice and sizes, but was not observed in tethered membranes, which is what the community calls just the regularly like covalently linked networks, which means there's something about this like, chainmail structure that causes it to curve at equilibrium. And you can kind of argue that this is like maximizing the free volume that each mini circle has to explore. But this got us thinking, or at least got me thinking, about the relationship between the topology of a like chainmail structure and the overall like shape of the network that it forms. So you can think about this not just in terms of like square versus hexagonal versus honeycomb or whatever, but there's a subtler point about the chirality of each of these links. So what I mean by that is if you have a chain of links, you can like twist it around any which way and its topology will not change. However, if you close the chain into a loop, then the total number of twists is something that is topologically fixed, and each number of twists and plus a closure makes a unique type of link. And this changes like sort of how the network behaves. This is related to like the twist writhe thing for DNA, if people are aware of that. So if you look at a square lattice, nope, got ahead of myself. So sort of around this time, like in the early 2020s, there were a few other simulations of kinetoplasts. One was from Luca Tubiana, and I'll talk more about this paper in the next section, but he was interested in how tension from the edge of the network causes it to buckle, and he imposed a constraint that did that. But he also noticed that the way he set the chirality of these loops in like a honeycomb, in the honeycomb lattice, changed how the network arranged itself. So he just randomized all that chirality and knew there was something interesting there. Around the same time, Tom O'Connor undertook what he described as his pandemic stress project, where he looked up different types of medieval armor and simulated them as polymer networks at different temperatures and looked at how they like curled up or folded or whatever. And unfortunately, this was never taken to fruition. So the only evidence that this happened is like the background of this like Carnegie Mellon marketing video. So I'm just mentioning this to tell you that I'm basically the third person to do what I'm about to tell you. I just happen to have a complete story about it. So I was interested in square lattice chainmail networks, and I'll explain why, hopefully on the next slide. I can't actually see the preview here. So if we have like a link connected to 4 of its neighbors, each link can go like over under, or it can go under over. And with 4 neighbors, there's different sequences that can happen. So this one goes over under over under. We'll call that alternating. This one goes over over under under. We'll call that non-alternating. And then the last one has a combination of like alternating and non-alternating. We'll call that one semi-alternating. And that's the one that's used in actual European chainmail. Called like the 4-in-1 weave. So if you kind of represent the connection between 2 adjacent squares, so first of all, you can tile a plane with squares. I think that's well known. And you can color the edges black for over under and white for under over. And with a square, you can make your unit cell have like black on the sides and white on the top and bottom, or you can alternate diagonally. And you can make these different like checkerboard patterns of alternating black and white to look at these different chiralities. If you do this with like the honeycomb lattice, which is how people think of kinetoplast topology, you end up with frustration. So you can alternate the lag there. You can alternate like white, black around one of these cycles, but then you have a point where you have to pick one to be the same color as its neighbor. So I was interested in how this choice of chirality influenced the networks, which is why I went with the square lattice. So to investigate these networks, I used 2 different techniques. One I'll call constrained gradient descent, or just knot shrinking. So I mentioned the rope length problem for knots, which basically asks what is the minimum amount of rope needed to tie a knot with a certain topology. This assumes that the radius of the rope is 1, and how that radius is defined I can get into if you care, but it's not the topic today. And just for example, the tightest known trefoil knot has a rope length of about 32.76, which means a 1-inch, a rope that is 1 inch in diameter, you need like 16.38 inches of rope to tie it. So there's a software called Ridge Runner that performs gradient descent on closed curve while respecting this no overlap constraint. And basically, I use that to you get the point by now. The other method is molecular dynamics, which basically simulates the motion of atoms under certain forces that you impose on the system. So in polymer dynamics, this is called a bead spring system. So you have beads, which represent the atoms, connected by springs, which represent the bonds. Their sort of motion is propagated into the future using the Langevin equation, which adds like drag and diffusion, which simulates a molecule in a liquid. at thermal equilibrium. So the forces involved are repulsion, basically almost hard sphere repulsion between atoms, which prevents them from going through each other, and finitely extensible springs. And just by tuning like the ratio of the repulsion diameter and the maximum spring extension, these simulations can preserve the topology of a knot or a link unless you let the ends pass through, like if you don't make it a closed loop. And this is all done in a software called LAMPS. So each method gets us different things. If we want to know how these molecules behave at equilibrium, then molecular dynamics gets us a picture of that. It's good for recreating experimental results. But the actual equilibrium structure depends on a balance of entropy and bending stiffness and self-avoidance. Whereas the rope length of a knot is a topological invariant, and you can think of like its minimal configuration as a like invariant configuration of that knot. Just gonna look at Ryan for reassurance when talking about knots. So when you shrink one of these chainmail networks, you basically find like what is the topologically invariant minimum structure of that network, which I think is useful for seeing where certain features come from. So basically, I simulated these with the molecular dynamics up to like 15 by 15 networks of 225 links, kind of parameterized to DNA, but more the DNA in the African sleeping sickness parasite, not the one that I study in the lab. And then gradient descent, it's a much less efficient algorithm. Also much more finicky. So I basically was able to go up to 5 by 5 networks with these 3 chiralities. So I'll show you the results first for the molecular dynamics. So with the alternating chirality, we get this positive Gaussian curvature. This is the one that goes over, under, over, under. And that's the same positive curvature we see in like actual kinetoplasts. So do I think that the parasite's life cycle is somehow having its DNA woven over, then under, then over, then under? Absolutely not. But if you did want to recreate them in a simulation, that might be a good way to do it. If you have the over, over, under, under chirality, you get negative Gaussian curvature, so more of a saddle or Pringle-shaped network. And then if you have the semi-alternating one, you get a network with 0 or approximately 0 Gaussian curvature. And this is the one that's used in actual armor. And that's good because if you put on your suit of armor and your chainmail has negative Gaussian curvature, that's not good. You just want that in your saddle. So I guess the main result here is that the chirality dictates the Gaussian curvature. So we can measure this basically by replacing each loop with a point at its center of mass and constructing a triangulation. And then like for each vertex in the triangulation, adding up all the angles and using the Gauss-Bonnet theorem to figure out the local Gaussian curvature. And again, we can measure this. We see it's positive where we expect, or where we kind of visually see positive. It's negative where we visually see negative. And it's close to 0 where we see flat until they start to get very big. And then well, then it gets complicated, but don't worry about it. We see the same thing with these height networks. So positive, negative, and 0 Gaussian curvature. Here we can see, especially in the flat one, they basically start to form like almost this crystal structure of how the different loops are arranged. So this, because we can only go up to 5 by 5, we can't see like, the whole like periodicity of the system. It's still a lot of edge effects, but we can look at the central and edge and corner pieces of the different networks, and we can see things like in the alternating ones, the corner pieces are basically bent into an 8. Whereas the non-alternating ones, the corner pieces are basically a stadium curve, which is the smallest possible curve that can have 2 other curves go through it. An implication of this is that you can have things that look like supercoiling in networks that are not supercoiled. Does everybody here know what supercoiling is? Okay, then it doesn't matter. All right. Another thing that's cool is that the thermalized graphene community is interested in what's called a crumpling transition. The idea is that a two-dimensional polymer, which is how they model thermalized graphene, is effectively flat at any length scale, but as these networks or these sheets get bigger, although they remain locally flat, there can be a loss of a global surface normal. And if you look at these networks, they eventually so they form a diamond shape, which I didn't mention. The diamond has about 2-to-1 aspect ratio, and it like wiggles only on the long axis of the diamond. So eventually, if they get big enough, they can fluctuate so much that sort of surface normal like here. You get the point. On the other side, basically no longer correlated, which I think is neat. So the reason I think this is interesting is because we have there's two broader like things from topology that you might think to describe this with. So with DNA, we have the Fuller-White-Caligariano theorem, which I guess also applies to twisted ribbons, but it relates the twist and the writhe to the topological linking number between either the two helices of DNA or like the two edges of a twisted ribbon. Whereas with surfaces, you have the Gauss-Bonnet theorem, which relates the integrated Gaussian curvature to the genus. So when we look at this system, the local chirality of the links reminds us of the Caligariano theorem. And the Gaussian curvature of the membranes reminds us of the Gauss-Bonnet theorem. So it feels like there should be something connecting these two for this particular system. And that might have some implications for broader systems. So we have a partial explanation from my collaborator, Mike Dmitriev, who's now at Texas A&M. He actually turned down a job at CSU San Bernardino, so he could have been local. Shouldn't have said that on the recording. Anyway, so this is the thing that mathematicians might find most interesting about this, and also the part that I understand the least. But I'll give a basic summary of his argument, which is that you place 4 circles in a square lattice, and you look at how you need to either twist and bend or incline and rotate, however you want to describe it, to make a closed loop in the space defined by those 2 angles. And when you close the loop in theta phi space, you can, in some cases, have a gap in Euclidean space. So there's a certain degree of non-closure caused by creating this loop in theta phi space. And to correct this non-closure, there's a certain amount of torque that needs to be imposed on the system. But because surfaces cannot have net torque, this is alleviated by causing Gaussian curvature in the network. And we can predict, well, he can predict what the Gaussian curvature should be. And we get the same positive where we expect, negative where we expect, etc. And if you find it interesting, there's the reference. So that kind of brings me to the end of part 2. Any questions on anything I've said so far? All right. So do we get kicked out of here at 1?
We don't, there's really nothing happening.
All right.
I think there was another meeting.
Oh, there is?
All right, check.
Well,
Yeah, we have a meeting still. There's a meeting.
Well, I don't think I'll be 25 minutes. All right.
There is a meeting at 1, yeah.
Okay, so the next part is called so the last part is called, are kinetoplast Borromean hypergraphs, and I'm going to again assume that not everybody knows what all those words mean, so I will sort of walk you through it. But just going back to how we understand kinetoplast topology, there was that experiment from the 90s. And then one of the first simulations of how these things form was done by Davide Nicoletto. It's kind of like the major player in kinetoplast biophysics over in the UK. Let's see if he's in here. Okay, he's not in here. Yeah, so he did this simulation where he had 50 ring polymers in a box, and they were allowed to pass through each other to form different link structures. And then he fixed the topology and used the Gauss linking number to compute the linking network structure. And you can see some of the things that form. And what he found was at the point where the network percolated, so you have a single like a single large component, that approximately happened when the average connectivity was 3, which is what we see in experiments. And this was like pretty early in the field. 50 is not that close to 5,000, but in several years later, next slide, please. All right, several years later, he and Luca, who I mentioned earlier, and some other people, they did what they made some very nice AFM images. And they developed what they call, like, I think, AFM steered simulations, where they just binarize their AFM image. Again, that's atomic force microscopy, just making those surface maps. They binarize their AFM image and then set up a simulation where each bit of DNA in that image is like an attractor for these rings. So they have the rings like anneal onto the measured structure and then allow them to pass through each other before locking it in place. And again, they use the Gauss linking number to compute the network structure. Again, they find that, on average, the mini circles have a connectivity of 3. This is like a graph structure that form. That's nothing like the honeycomb lattice. And they observe mostly Gauss linking number 1 for linked molecules, occasionally Gauss linking number 2. If you don't know what that means, I'll explain it on the next slide. And I'll just say this is the best paper that's been written on the topic. It's better than mine. So if you're interested, you should read it. So the Gauss linking number is an integral or a double integral over 2 curves that calculates how many times one curve passes through another. You can see examples of like linking number 1, 2, and 3 up there, but there are a few links which have a Gauss linking number of 0. So this one is called the Whitehead link. And so even though the orange and blue are linked, you can kind of think about how the flux of blue through orange is the same both ways, so it cancels out the orange twist in the middle. More commonly known are the Borromean rings. So these are 3 linked rings where no 2 linked rings are linked to each other. So you see orange or red is on top of green, blue is on top of red, and green is on top of blue, but you still can't pull them apart. But if you remove one, the other two can be separated. So you can extend these Borromean rings into bigger structures, which are called Borromean chainmail. And probably the first picture of this was drawn by James Clerk Maxwell in 1877 on a postcard. A couple of years ago, I saw this picture of Borromean chainmail on Wikipedia, and it just nerd sniped me in a very big way. So I've had a lot of questions related to that since then, which I've been trying to investigate and currently am with Ryan and Jonathan. So looking at this type of structure, there's a few questions we can ask, like if kinetoplasts had Borromean connections in them, how would we actually know? And I will just say that this is not a burning question for the treatment of parasite diseases. It's more, my own curiosity. But generally speaking, what are the properties of Borromean networks? Like when I first saw these, I was interested in like the percolation of these kinetoplast networks. So how would something like that undergo percolation as you remove circles? You can see like if you remove all the blues, I think all the reds will float away. But what if you remove circles randomly? And then all these simulations are done by putting just random rings into a box and letting them link. We'll call this an Olympic network. Can you randomly form Borromean networks within a broader Olympic network? We don't, or at least didn't know. So the challenge is that you can't use the Gauss linking integral to detect Borromean rings. There's a few other things that in principle can do that. So there's something, the extension of the Gauss integral to more loops is called the Milnor triple integral. And a lot of people prove that this works, but not a lot of people, perhaps 0 people, have made an algorithm that can actually do this. So there was a group about 12 years ago that claimed they could, but I have not been able to replicate their paper and nobody else has tried. But there's a knot invariant called the Jones polynomial, which can detect Borromean rings, and there's a software package called Topoly that if you give it the Cartesian coordinates of 3 closed curves, it will tell you what knot or link those curves make. So calculating that for like one of these like simulated kinetoplasts can be challenging, not just because the knot invariants are challenging to compute, but because of the combinatorics. So if you have 50 loops, you have 1,225 possible pairwise links, but then you have 19,000 possible triple links. So just setting up a system like the one that Davide did his first study with 50 loops, it took about 3 seconds to compute the Gauss linking number between every pair of links, but then 7 minutes to check every 3 links to check their Jones polynomial for Borrmean linking. So with 5,000 loops, there's 12 million pairwise links and 21 billion triple links. So you can maybe argue that I didn't give this a fair shake and didn't really optimize it, but it's not really feasible to do this at this time using the Jones polynomial. So about a year ago, I had a bit of a eureka moment when I remembered seeing these Borrmean rectangles. You can see like these rectangles form Borrmean rings. And a fun but irrelevant fact is that if you have 3 golden rectangles in Borrmean rings, they're corners from the vertices of a regular icosahedron. But more importantly for this topic, you can determine whether these are Borrmean linked just by looking at like the relative positions of each corner, assuming that they're all in 3 perpendicular planes. So if you have 2 rectangles, they kind of have 3 ways of orienting around each other. Either they can be spatially separated, they can have a Gauss linking number of 1, which is called a hop link, or one can go through the other, which I will call piercing. So if you have 3 rectangles, all with mutually perpendicular normal vectors, and A pierces B, and B pierces C, and C pierces A, or the reverse, then they form Borrmean links. And this allows us to calculate or detect Borrmean links in random rectangles just by looking at like the coordinates of each corner. So kinetoplasts are typically represented as graphs, at least when we look at their topology. So a graph has an edge that connects 2 nodes. With Borrmean rings, we require 3 edges and 3 nodes to define a connection. So that makes it a hypergraph, which is a generalization where each edge can connect a certain number of vertices, in our case n. So we represent these basically if 2 rectangles or any 2 closed curves form a hop link, then we represent them in a graph by sharing a blue edge. And if any 3, such as the red, the yellow, and the blue here, form a Borrmean link, then we represent them as forming a red triangle in this hypergraph. And if we remove one of the edges or one of the nodes from the triangle, then the other 2 edges vanish because we no longer have Borrmean links. Each graph is equivalent to a matrix called an adjacency matrix. So if something is, so we just can represent this, each blue edge as a 1 in the matrix, each red triangle as a bunch of 2s, and these matrices are symmetric if the graphs are undirected. And so to answer the question, are there random Borrmean links in random Olympic networks? All we have to do is generate a bunch of random rectangles in a box and then check every perpendicular pair for linking. If they are linked, we add a 1 to an adjacency matrix. If they are not linked, we see if they pierce each other. And then for every 3 mutually pierced rectangles, we know that's a Borrmean link, and we add 3 2s to the adjacency matrix. Then we convert that to a list of connected components. So, and this here's just an example going from rectangles in a box to the adjacency matrix to the hypergraph. So with the Jones polynomial, 50 ring polymers took about 7 minutes. With the rectangles, 1,000 rectangles took about 2 seconds. So we can learn things that we can't really learn with the polymer simulation at this time. So my motivating question was, can Borrmean links randomly form in stochastic Olympic networks? The answer is yes. So here's a random example where I've just colored, or I've made every rectangle that has a Borrmean link bold. You can see there's a bunch of bold rectangles through there. Here's an example of a hypergraph where we see red triangles sprinkled throughout, but if you make it denser, then you can have a percolating network of Borromean links within this broader network of hop links, which I think is neat. Another interesting result, if you have congruent rectangles and you end up with two different Borromean hypergraphs that do not share any Borromean connections, one in which all the rectangles are landscape and one in which they're all portrait. These can be connected by other, like just by regular hop linking, but not by Borromean linking. And the way to think about this is if you start with two identical rectangles and you rotate one of them out of plane, they can no longer pierce each other. They can only either be linked or intersect in 2 points. Whereas if you rotate one of them in plane first, then out of plane, then you're able to have this piercing. But if you allow their size or aspect ratio to vary, then you no longer have this separation. So actually, let's see, do I want to talk about this? So if you're interested in the percolation threshold of Borromean networks, it's already proven that a hypergraph will have a higher percolation threshold. So what I mean by a percolation threshold, for example, if you have like the square lattice of ones and zeros, you need 59.27% of them to be one to have like a single big component in your network. So we can measure the percolation threshold of any type of network if we define connectivity. Below the percolation threshold, almost no component is part of the biggest component or the biggest cluster. Above the percolation threshold, almost every component is part of the biggest cluster. So we just define like 50% of rectangles being in the biggest structure to define percolation. What is interesting, and I have a partial explanation for, but won't get too into, is that if you look at the hop linking percolation threshold, it is roughly independent of the aspect ratio of the rectangles. If you look at the Borromean threshold, as they get closer and closer to squares, you need a higher and higher density of rectangles to form a percolating Borromean network. But as they get longer, it's easier for them to pass through each other. And that eventually plateaus at about 10 times the critical density for regular hop percolation. So this was initially motivated by kinetoplasts. So a kinetoplast has about 5,000 components. They have an average valence of 3, and they are approximately 2-dimensional in their network structure. So we can just generate a bunch of rectangles in a disk instead of a box. We can tune the radius of the disk until it has an average valence of 3, and we can look at the network structure that forms. Here's an example. This one only has 3,000. And we can see that there's various red triangle clusters dispersed throughout. I just have a picture of some of the biggest ones and therefore the most interesting ones there. But if we can see that when we tune the density of these networks with 5,000 rectangles, such that the average valence is 3, then about 1,000 rectangles participate in some kind of Borromean linkage, which is a pretty high percentage. So if you believe that this is somehow analogous to these DNA structures, that might be imply that there's a lot of Borromean links within kinetoplast DNA. You might think this system is too abstract. So to see whether that is even possible to detect experimentally, we can estimate what happens when you randomly remove like molecules from the network or rectangles from the system by cutting DNA with an enzyme or just deleting a rectangle. And if you ignore the Borromean connections, you will overpredict how many single components are released as the network dissolves, and you might underpredict how many triple components or double or triple components. No, I think you'd still overpredict, but there's an experimental prediction based on whether Borromean links exist or not, which in principle could be detected, but I don't think by me. So this is actually my sort of second investigation into Borromean networks. The first one Ryan was involved with, I didn't talk about because I thought this was more interesting. So these rectangles are still fairly removed from the actual experimental system. So now with Ryan and Jonathan, we have, mostly they have developed a way to determine whether any three like arbitrarily oriented ellipses will form Borromean links. And we can do the same type of investigation with a system that is a bit closer to something that could be realized in a molecule, and also just learn some neat things about networks of ellipses. And then eventually we hope to extend this to actually understanding kinetoplast networks. So just to summarize, so kinetoplasts are weird and cool, and if you didn't know about them, now you do. And I've talked about these studies of link chirality versus Gaussian curvature and random Borromean percolation. What I hope is that this is more than just like some cool facts about a specific system, but that we can maybe extend this